Hi guys, welcome back to Bright Future YouTube channel. My name is Kwadriasha Omba Shabanzila Shankangatela Rijweni. Nimduamba Mina Chavinchumundi Chabala Maika. Thank you so much for tuning into this video. So if you haven't subscribed, please do subscribe. Comment if you have any questions, and then like and share with all of your friends on all of your social media platforms. If you're in need of a private tutor, you're more than welcome to send an email or a text message or a phone call to the numbers on the beginning and the end of this video. So in today's session, we're doing life sciences. We are answering March test 1 question paper of 2020. I opted to leave question 1 because it asks questions that we have not yet covered in this sessions. It doesn't cover nucleic acid and meiosis, so I opted to go for question 2 and question 3. So let's start with question 2, which is section B. We are given the diagram here of a nucleic of a phase of meiosis. The diagram represents the phase of meiosis, so we need to analyze this diagram and then be able to answer the questions that follows. Let's go. It says identify A and B. We go to our diagram. A, it's the nuclear cell membrane or a plasma membrane, and then B, it's the centrosome or central. So one of the two in here is the correct answer. You could either choose this or this or this or that. And then it says identify the phase of meiosis. I've already answered and said this is metaphase 2. So as you can, if you can remember or recall from my previous lesson sessions, I have shown you how to identify meiosis 1 and meiosis 2 division. So you should be able to answer the questions. 2.1.3 says describe the process responsible for the appearance of chromosomes as shown in region X. So let's look at region X. Region X shows you a chromosome that had an exchange of genetic material. As you can see in this one, it's not colored all of it, but it's colored at the tip, and in here it's colored everywhere, but it's not colored at the tip. So this is evident that there's a genetic variation. So we need to describe that process. This X basically points here on the dotted section here, the one that's dotted. I just didn't extend the line to the dots. So the answer to that is it's crossover. It occurs in prophase 1. Chromatids of homologous chromosomes cross at a point called the chiasma to exchange genetic materials. So this is how you answer your questions. So you can check on the points depending on the number, the mark allocated next to the question. And then let's go to 2.1.4. State two significance of meiosis. If you can recall from the previous session of meiosis, so you can recall from the session that I did previously of meiosis, where I told you that it's important for you to know the significance of meiosis. So in here we were asked to mention 2, but I mentioned 3, and it's okay. You can choose any that you can remember in an exam. It's production of gametes, prevents the doubling effect or during fertilization, introduces genetic variation. Now let's go to 2.2. So this was the diagram that was drawn. I tried to draw it, but I didn't finish. As you can see, 14 to 20 doesn't have chromosomes, but they were the same as the one for 13. So I was like, it's okay, I don't have to continue. And here there is chromosome 11, which is labeled A. So let's answer the questions below. State the correct biological term for the chromosome pair number 23. So pair number 23 is this one, and the answer to it is gonosome. So let's move on again to 2.2.2. Identify A in the diagram, which was chromosome number 11. It's called the homologous chromosomes. If you go back to the sessions of meiosis that I've recorded, you can actually see what homologous chromosomes are. I did explain to you. 2.2.3 explain the consequences if chromosome pair number 21 in a human cell fails to separate during meiosis. So what happens if it fails to separate? Its non-disjunction during anaphase 1 results in abnormal gamete with an extra chromosome being formed. The fusion between an abnormal gamete, 24 chromosomes, and a normal gamete results in an individual with 47 chromosomes instead of 46, leading to Down syndrome. You can go, when you're answering this question, check the mark allocated to it, and then you're gonna answer based on the mark allocation. Let's move on to question number 3. So for question number 3, the diagram below shows some codons of mRNA together with their corresponding amino acids. So now this is mRNA. Now the base sequence of a section of tRNA used during translation is shown below. So now this is tRNA. Let's answer the questions. How many different types of amino acids are required for tRNA strands during translation? It's 5. That is my answer. Now, write down the complementary base sequence of the DNA strand that formed anticodon 5 of the tRNA. It's ATG, and I'll explain to you why I'm saying that. 3.1.3 explain the effect on the composition of protein molecule if the base sequence at 4 was AGU instead of ACA. So the answer is that both AGU and ACA carries serine. It will be replaced by another serine, which means you'll have the same protein formed. Let me show you how. They say if it was AGU instead of AUG. So when you complement this, you form an mRNA. mRNA will say it's, for mRNA it will be U, G will pair with C, and U will pair with A. So it was gonna be UCA. So UCA, it's serine. As you can see, it's UCA. It's serine. You pair this to the mRNA, and then it will show you which amino acid it will form. And then for number 5 as well, it would be U, A, and C. So no, it's U. Yes, it's U. A, is it 4? I think it's, no, it's 4. I'm sorry, it's 4, not 5. It's 4. So it's U, A, it's U, G, U. So this will form UGU, which is UGU serine in this case as well. So it will form the same even if it was to be replaced, because they both hold serine. Let's move on to the next question. We are asked to describe the process of translation. On my previous session for nucleic acid, I did explain that you guys need to know the process of translation, as well as the other processes, because it's 2 of them. So I did tell you that sometimes they give you as a diagram, sometimes they want you to describe it. So you should be able to mention all these points depending on the mark allocated. So as I was saying, for question number 3.1.2, I want to show you how they got to ATG. So we were asked to identify the DNA that was used to pair to mRNA, which got used to pair to tRNA, which was number 5. So let me show you an example. If you are given UAC, which is your tRNA, and they want you to know what DNA codon was used to form tRNA UAC. So you have this that you given, you pay it to the mRNA. When you pay it to mRNA, U paired with gets paired with A get paired with U, and C get paired with G. Now you have your mRNA. So you want to know as well what mRNA you need. Well, what you need to know, you need to find your DNA that was used to pair with mRNA, this mRNA which formed this mRNA. So it's like you're going back in reverse. So what was used was A paired with T, U paired with A, and G paired with C. Again, here is your DNA, and then you go back to pair it again. So your T gets paired with A get paired with T, and C get paired with G. So this is how you know your complementary, your DNA. So you have 2 DNA strands here, so hence you have to go again for the second DNA. So it's the answer is this one. I hope you guys understand. So what I'm basically going to do is to end this session, because this is the last part of nucleic acid. So if you have any questions, you can put it on the comment section below, and then I'll be happy to answer it. Thank you so much for tuning into this video. Please don't forget to subscribe, like, comment, and share to all your social media platforms.